Hi, so today we're going to talk about Hershey-Chase Experiment, and this experiment was set to help confirm that DNA instead of protein is the genetic material. And so this experiment has utilized this type of virus, which only targets bacteria cells, and so this is one of my favorite type of virus just because of its super cool structure. And by the time of the experiment, it's already known that this virus it's composed of protein code along with the DNA that's being included inside. And so starting this experiment, they firstly created two strands of bacteriophages. The first strain they basically they use radioactive material to tag the protein code of the bacteriophage, and the second type they use radioactive material to tag the DNA inside of the bacteriophage. And by allowing the two types of viruses to infect the bacterial cells and allowing the bacteria to replicate, carrying this genetic material being transported from the bacteriophages into the cells, and finally through centrifugation, which was able to separate bacteria from the viruses because of their differences in mass, and they were able to see that whether the protein or if it's the DNA that's been left in the bacteria. This is important because imagining to allow a bacteriophage to replicate, it is important that the bacteriophage at the first step was able to transport its genetic material inside of the cell and use the cell's machinery to express the viral protein, and hence to make more of its the same kind. And what the researchers has found is that the DNA is being left in the cell, and hence it was able to support that the original hypothesis that the DNA is the genetic material instead of protein.